Alright guys, last time we left off with the previous video talking about the different types of hormones that are in your body. They can be a peptide hormone, like a protein type hormone, like insulin, glucagon, leptin. Those are all examples of, or epinephrine, those are all examples of peptide type hormones. They could be amino acid derivatives, which are individual amino acids, or they could be in the form of a steroid, which has a cholesterol base or precursor to it. They're a ring structure. They're completely lipid soluble or hydrophobic. They a lot of times have to be transported using a protein to get them around, because remember, lipids are nonpolar, but proteins are polar, so they need those proteins to be able to mix in our water environment. Now we're going to take a look at some of these different mechanisms for causing change using hormones to signal. We're going to look at a steroid hormone action, and then we're going to look at peptide hormone signaling. First one let's take a look at is steroid hormone action. This is an animation from McGraw-Hill. It's actually the same company that created the text for this course, which I know we haven't used a whole lot, but that's okay. If you take a look at this, we're not going to memorize every single component of this, but we're going to use this as a framework to base an understanding from, to get an idea of how these signaling mechanisms work and these biochemical processes or cascades or chain of events occur to cause change in your cells using the endocrine system.
Steroid hormones are not water soluble. They travel in the blood attached to protein carriers.
Now remember, steroids are non-polar. They can't mix with polar substances, so therefore in order to make them mix, they bind with proteins, and sometimes they're called lipoproteins because it's a lipid substance combined with a protein, and that protein just shuttles it around and makes it soluble in the water so it can be transported.
When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell.
So again, dissociate, I'm hoping you can make an inference there. That just means breaks apart, comes away from, releases from. And then if you notice how it went across the membrane, there was no protein involved in getting it across, because remember, it's nonpolar. So it has the ability to slip right between these phospholipids and through passive diffusion. Passive meaning it didn't require any additional energy, and diffusion and slipping from high to low concentrations. Again, passive also refers to not needing a protein to get across either. If it was facilitated diffusion, it would have had to go through a protein here. But this is lipid soluble, it can slip right through.
Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown.
So in other words, what they're saying is, if this animation showed that, it would just show the little circle with the S. It would float all the way down in here and then combine with the purple thing to form a complex. All right, pay attention to this, what happens next.
The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA.
Now understand, to make it a little bit easier to understand, they've omitted a ton of different enzymes that are going to be involved here. But because of this certain molecular shape and the other enzymes involved, it specifically attached to a certain region of the DNA molecule, and it's going to start or initiate or promote transcription. That just means they're going to make a copy of the instructions to be used out in the cytoplasm to build a protein from. So watch this.
These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site.
So anytime you hear transcription, they're talking about making a copy of the DNA so that it can be used to build a protein, because that's what those that little ribbon-looking thing there. That's an set of instructions on a certain protein. Which amino acids need to be arranged to build the protein.
Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
So what you got right here is a ribosome here that is just sliding along, and it doesn't show because it'd get really complex, but there are ribosomes, there are amino acids being brought by other RNA molecules called tRNA molecules, and basically just linking them together and creating proteins. All right, this would be the primary structure of amino acids being linked together based off the instructions that were copied because of this hormone. So that hormone came in, diffused across, went to the nucleus, it binded, whatever the correct form of that word would be, to that specific spot on the DNA and caused that section of instructions to be copied, sent out, and then used to build a protein. And you can see there it folded into its active shape. And now whatever that protein is supposed to do, that's carrying out gene expression. If it's supposed to build a structure, it'll build the structure. Or if it's supposed to cause a change inside the cell, maybe it's an enzyme to start another reaction inside the cell that is necessary. Whatever it does, that is gene expression, because this gene that was copied is being expressed when the protein carries out its function. All right, let's take a look at this next. So that was a steroid hormone. Let's take a look at a peptide hormone. Now, most of everything we need to see the difference occurs in the first 15 seconds of this. It gets kind of hairy towards the middle, but understand we're looking at the big picture here. Right? We're not going to memorize the individual cascade steps. I just want we're going to use this as a framework to base our knowledge off of, as far as getting a general idea of how these hormones communicate and cause change.
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and
that means it's lipophobic. It doesn't like oils or fats or nonpolar substances. So it does float around freely in the water environment. If you notice, it doesn't have to have a protein to carry it around because it's already soluble. This is a lipid, or excuse me, a peptide hormone.
And therefore unable to cross the hydrophobic plasma membranes of its target cells.
Because it's hydrophilic, it's hydrophilic because it has a charge to it. It's polar, so it can't bounce through. It has to go through a protein to get across.
Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated.
Okay, so making this a little bit easier. This thing binds to here, causing these proteins to combine with this and become active. It doesn't show it, but there are going to be chemical bonds rearranged, and that converts this protein complex into an active form so it can carry out the next phase of the reaction.
Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2.
Break apart.
The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the...
Okay, GDP, GTP, those are both energy storing molecules, very similar to ATP and ADP, which you may be more familiar with. So on a biochemical level, GTP is very, very similar to ATP, and GDP is a lower energy version of similar to ADP. So again, this is the higher energy version, just like ATP. This would be the lower energy version, just like ADP. It has one less phosphate.
Subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of CAMP from ATP. The CAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins.
Okay, I know that's a lot of biochemical gibberish right there, stuff that we don't even need to get into for this unit, or this course, mind you. But I want you to understand, get the bigger picture here. When this epinephrine comes in contact with this specific shape-dependent receptor, it causes a chain reaction of events to occur. That binds there, that one breaks apart, a more energy-rich form of that molecule attaches to it, it kind of diffuses over, it has to combine with this substance over here, all right, which then uses ATP to create these other what's called cyclic AMP, or cyclic adenosine monophosphate. Only has one phosphate group on it, which then that substrate or that product goes and binds with this protein, creates a protein kinase. And as it mentions, kinases are specific in their kinases are a group of enzymes that just add phosphates to things. They phosphorylate things. So watch this.
Phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate.
So time out. All that had epinephrine attached here. This thing dissociated or broke this part off. They traded a GDP for GTP, bound with this, created the cyclic AMP. I guess I can show you all this. This thing breaks apart, trades it for a more energy-rich version, creates the CAMP, which binds to that, which adds a phosphate to that, which then causes glycogen to be broken down into glucose. So again, all of this chain reaction and production of glucose would have never happened had epinephrine never bound to its receptor. So again, just comparing steroid hormone signaling, steroids, since they are lipid soluble, they can diffuse all the way through the membrane and then they interact with the receptor, whereas peptide hormones, their receptor, they actually never enter the cell. They just dock in or connect with a receptor on the surface of the membrane, and then they transmit signals through the membrane via that protein to cause a chain reaction of events involving several other proteins and pathways within the cell. So again, that's the big picture idea. I don't want you to try to get confused and memorize all this different stuff going on. Just understand, if that peptide hormones, their receptors are on the surface, then the signal is transmitted through that receptor protein to the inner workings of the cell, causing a chain reaction of events, a biochemical cascade, because again, that has to bind, activate that, that binds with that, activates that, this CAMP binds with that, which activates this, and then the phosphorylase, its job is to add phosphates to this glycogen molecule and cleave it, hydrolyze it into glucose to be released whenever you need energy during a fight or flight situation. So epinephrine, your adrenaline, that's how it causes your body to dump glucose into the bloodstream, and this all happens pretty fast. But we had the luxury of this video to help slow it down for us so we can kind of take a look at it. If we were studying the intricate biochemical processes of epinephrine signaling specifically, we might look at some of these intermediate reactions more closely. But right now, it's just to give us an idea of how hormones signal and cause change. Let's see. I've got a couple of diagrams here. These two again show you just steroid. It's rather than animation. It shows you the steroid diffuses through, comes in contact with the receptor, goes to the hormone respondent element on the DNA. Basically, it attaches to a specific spot based on its shape, and then it basically causes that DNA to be copied, making mRNA. The mRNA is then read using ribosomes to make proteins, and then those proteins go and do what they're supposed to do, and it carries out its function and expresses the gene. So whatever this hormone was intended to cause, it's going to cause it through the transcription and creation of certain proteins that carry out or amplify the signal, as it mentions right there. So again, that's just steroid hormone signaling. Here's a side-by-side comparison. As you can see, the secretory cell releases the hormone into the blood, goes to a receptor in the membrane. This would be a peptide response. This over here would be a typical steroid response, because you can see it goes into the cell membrane. This one actually goes into the nucleus before it actually starts causing a change in transcription to occur to create proteins. So again, that's kind of a rundown on some examples of how hormones cause change and signal each other in the body. Hope it helps.